Good morning. My name is Miguel Velez. I'm a freshman at the University of New England in Biddeford, Maine, majoring in medical biology. Formerly, I was a dental assisting student at Montachusett Regional Vocational Technical School, where I worked with my fellow researchers here to put this experiment together.
Good morning. My name is Russell Hulbert from Fitchburg, Massachusetts, and I am a junior in the Graphic Communications program at Monty Tech.
Good morning. My name is Brittany Velez, and I'm currently a junior in the dental assisting program at Monty Tech.
And unfortunately, our fourth member, Victoria Hulbert, is not able to be with us today, but she is a freshman at Norwich University in Vermont.
And the title of our experiment is The Effects of Arthrobacter on Polyethylene Decomposition Rate in Microgravity. So our big question, would plastic-eating microbes called Arthrobacter bacteria decompose polymers at a quicker rate in microgravity? And based on a few things that we knew, we were able to propose our hypothesis. So the first of these is the majority of microorganisms thrive in space. We were able to figure this out and observe this in our last experiment that went aboard Atlantis STS-135, where we sent up Lactobacillus acidophilus bacteria into space, and were able to see that they did reproduce at a greater rate. Also, there have been studies done that show that bacteria like E. coli also reproduce at a greater rate in space. And second, there are bacteria capable of degrading plastics. We had a doctor named Dr. Mincer who helped us with our experiment with finding out the results with it, and he was great because he's actually currently studying Arthrobacter bacteria on plastics at this time. Because there's plastics in the Atlantic Ocean, and these Arthrobacter bacteria are breaking down those plastics, so we're able to see that that's actually taking place now, and that would help us to form our final hypothesis, which is microorganisms should be able to degrade plastics at a faster rate in microgravity.
Now, many of you may be wondering how this knowledge is beneficial. Well, at first, we kind of went to the large scale and thought, okay, well, because Arthrobacter bacteria can decompose plastic at a faster rate in microgravity, then maybe we could recreate a microgravity environment here on Earth where we could just simply send all of the pollution that is plastics and it could be decomposed at a quicker rate. However, we came to our senses and realized that that's not really cost efficient, and at this time we don't really have the technology to do that. So we started to think on a smaller scale, and we thought about the waste products that are produced on the International Space Station and how Arthrobacter may be able to decompose these plastics at a faster rate rather than sending the waste products produced on the International Space Station back to Earth, which is kind of costly. You could simply decompose them in the International Space Station, which could be beneficial. There also may be a byproduct that is produced as a result of this event, and this could also be used to create a substantial future.
Okay, so how do we test this? The type of materials that we used was, as you guys can see, a type 3 FME main volume. So this was the plastic tube on the outside that housed both the short ampule A, which was a glass tube, and short ampule B, which was also a smaller glass tube. And what we did was we took the diatomaceous earth, which is the medium that the bacteria would use to continue to grow and reproduce, and we put it into that glass ampule. And the short ampule B held the freeze-dried Arthrobacter bacteria. So what the astronaut would do while they're in space is break both of these glass chambers, causing all three mixtures to come into effect and allowing the experiment to take place. Because in the main volume there was nurdles, which are small plastic polyethylene plastics, so that would all mix together and then the experiment would take place. And that took place 23 days into flight. And the way that we kept everything sterile throughout our time in preparing the experiment was by using isopropyl alcohol and also keeping everything underneath a fume hood. And finally, to keep everything sterile, we had gloves on throughout the entire time of preparation.
Okay, so it took us 2 attempts, but we were finally able to analyze our results. Thanks to Dr. Mincer and his team at Woods Hole Oceanographic Institute, we were able to use an electron microscope to analyze the nurdles that were exposed to both microgravity and our ground truth, which was a really awesome experience. Like, that was really cool. Here we have images of our control and our microgravity sample. You can see that little green line down there. That is a scale of 20 micrometers. So that's how close we're able to get. You can see on the control, it's a very rigid surface, something that feels smooth to the human hand, as many cuts and bumps to it. And what we were able to identify as colonies were these white clusters on our microgravity sample. These colonies, we identified as them because there were none on the control sample, so it was very conclusive in that regard.
After analyzing our results, we kind of questioned whether or not the bacteria actually was able to survive on our ground truth sample. So in order to make sure that they actually survived, we did a DAPI stain test, which is a DNA nucleotide stain test, which measures the volume of the bacteria. And as a result of this test, we did find that bacteria was actually present on both the ground truth and our microgravity sample. And when looking at our computer microscope images, you can see that on our ground truth or control, you can see like the lighter spots are areas where there's clusters of Arthrobacter, so they were present. And on our space or microgravity sample, you can see that there are also those similar lighter spots, which signifies that there is Arthrobacter present. However, you can see a difference in the microgravity sample where these clusters are much larger and the chains are longer, the rod-shaped chains are longer, as opposed to our ground truth sample.
So based on these two tests that were conducted on our experiment, we were able to find strong evidence that Arthrobacter will colonize and potentially degrade plastics in space. The fact that Arthrobacter grew in both mediums shows that the conditions we created were favorable enough, and the fact that there was only visible colonization on the surface of the polyethylene on the microgravity sample shows that given enough time and probably proper growing conditions, we could facilitate and possibly observe the decomposition of polyethylene. So based on the study we conducted, some future studies would be what conditions are best for Arthrobacter and what byproducts could result from biodegradation of polyethylene, as Brittany had said. Thank you very much. Do we have any questions? Good morning, sir.
Hi, good morning, guys. I have a question. If this is a benefit for a space flight where they could decompose as plastics, could it also be a problem for future astronauts or future colonizations in space?
What was the last part of the question? I'm
Could
sorry.
it also be a problem for future colonizations in space or spacecrafts where it will decompose some plastics on the actual spaceship?
That could present a problem. You have a very good question. It very well could, but in the matter of space, I think a lot of situations could go wrong if not properly facilitated and properly contained. So I think if conducted again and if ever applied, it would have to be done in a properly controlled environment.
Thank you, guys.
Did you use a specific species of bacteria?
Well, through our work with Dr. Minster, we were able to identify this Arthrobacter, but we only used the genus. There was no direct species that is observed to be responsible solely for this process yet. So in all the experiments that Dr. Minster had done and in his discussion with us, he strongly recommended that we only use the genus Arthrobacter.
Good morning, great job.
Thank you.
I'm curious how you got in contact with the researcher, the scientists who helped you with this part of the experiment. It's been very, very beneficial for our team, and I was wondering how you guys made that connection.
Well, through the initial research, once we were trying to brainstorm our project, we really just found an article on the internet, and it had his name and a contact. And so through help with our awesome facilitator, Mrs. Paula DiDiego, we were able to contact him directly, and he was more than willing to help us with our studies and help us with our experiment.
How did the glass not break when it was being flown up into space?
The part of the procedure for conducting our experiment, there were 2 glass carbules within our experiment container, and they had to be cracked in order for the experiment to be activated. So they did have to be cracked. So yeah.
So did you guys stop the reaction when it came down from space, or did it just continue going until you guys received it?
It continued going after we'd received it, but we had we had analyzed fairly close to the actual event, because what we wanted to see was the growth, and the colonization is what we really wanted to observe.
All right, thank you.
Why were you purposely putting mold into space?
Purposely putting what in space? I'm sorry.
Why are you basically killing plastic with mold? Because you would want to kill mold too.
So why are we getting rid of plastic is your question?
No, why would you put mold into space on purpose?
Well, it isn't technically a mold. A mold is technically more of an algae, protist kind of animal, but bacteria, usually humans associate the word bacteria with negative implications. But this bacteria, it's actually found in the soil. It's a fairly common strand of bacteria in most soil samples, and it has no harmful effects on humans as so far detected. Thank you for your question.
Great job, guys.
Thank you.
What is the how economically efficient would it be to break down plastics in space as opposed to the process that they have currently to get rid of waste? Using your new method, what's the economic implementations of it?
That is, do you want?
I haven't personally researched the cost exactly. I'm not sure if one of them have, but I could just imagine the cost of flying out to space and then back with all that jet fuel cost. So you can just imagine not having to make that extra trip, which I'm sure is thousands of dollars in jet fuel, and having to send these men and women up into space is just a lot of money. So being able to do this in one process rather than having to do multiple trips, I'm sure is very cost effective.
Thank you.
Thank you.